Hi, everyone. I'm excited to have you here for episode 833 of People Behind the Science. I'm your host, Dr. Marie McNeely, and this week we are rebroadcasting our interview with Dr. Kristen Lynch. Listeners, Kristen's research focuses on alternative splicing, and in particular, how cells decide when to use an entire portion of their DNA instruction manual versus when to exclude parts of the instructions that either aren't useful or would be harmful. In particular, Kristen is interested in alternative splicing in the immune system and what happens when cells are faced with an immune challenge. And she shared some fantastic stories from her life and research in our interview. So I hope you enjoy this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of the People Behind the Science podcast. Today, I am thrilled to introduce you all to Dr. Kristen Lynch. So, Kristen, thank you so much for joining us on the show today. How are you?
I am fine. Thank you very much for having me.
Wonderful. Well, we are excited to learn more about you and your work. But to start off with, I want to take a moment to introduce you to our listeners. So, listeners, Kristen is a professor and chair of biochemistry and biophysics at the University of Pennsylvania. She received her bachelor's degree in biochemistry and her Ph.D. in biochemistry from Harvard University. Afterwards, Kristen pursued postdoctoral training at the University of California, San Francisco. She served on the faculty at the University of Texas Southwestern Medical Center prior to joining the faculty there at the University of Pennsylvania in 2009. Now, Kristen is the recipient of many awards and honors for her work, including a National Science Foundation Career Award. And listeners, Kristen is here with us today to talk about her experiences in life and science. So, Kristen, we want to get to know you today, both as a scientist, but also as a person. So can you start by telling us what you like to do when you're not busy doing research?
I am an avid outdoors person. So when I'm not doing research, you can usually find me on a kayak or a paddleboard or a bike or a hiking trail. And if the weather is bad, a yoga mat.
Excellent. So what kind of yoga do you like to do?
Vinyasa, mostly.
Okay.
In fact, we have a yoga class later today that we run at UPenn with the medical students, which is a lot of fun.
That's awesome. Well, it sounds like you're staying active there, staying fit, and having lots of fun getting outside. And this is a great time of year to do it, I think. It's not quite cold enough yet to keep you indoors.
Yeah.
So now, can you tell us a little bit about your work next, and specifically how you describe it to someone who may not at all be familiar with your field?
My field, or the question that really interests me in my lab, is one of alternative splicing. So what that really means to a non-scientist is that each cell in our body has the same instruction manual, if you will, and that's the DNA. But each cell uses different chapters of that instruction manual. The decision of which chapter to use is the field of transcriptional regulation. But even once a chapter is decided to be used, different cells and under different conditions might decide to only use part of that chapter or to get rid of pages, if you will. And that process is called splicing, so the removal of certain information from a gene. And what we're interested in understanding is how cells decide essentially when to use the whole chapter, when to use only part of the chapter, how to get rid of those parts of the instructions that aren't needed or would be deleterious for certain environmental conditions.
Very cool. And I love how you use this book analogy to really make it accessible. That's awesome.
Thanks.
So now, can you tell us a little bit next about what motivates you? I think as a scientist, there's a lot to do in a day. So do you have a favorite motivational quote or a saying that really keeps you going, Kristen?
My favorite motivational quote is "fake it till you make it."
I love it.
The reason I love that is I think there's a lot of insecurity in science. That is, we all are striving to do great things and learn how this world works. And that's a daunting task. And I think it's quite easy for people to feel, myself included, that we're not up to the challenge. And we look at people around us who are tremendously smart and capable and successful and think we can't possibly live up to that level of excellence. And what I've realized over the years is that we're all insecure and we all think that we're perhaps not as good as the people around us. And the people who are really successful are the ones that have not let that be paralyzing. And really what that takes is to believe that what the other people around you are saying about your abilities is maybe more true than the negative voices inside your head. So I tell my students that you really do have to maybe pretend that you're more capable than you feel, because you really are that capable. And there's merit to faking it until maybe you believe yourself.
Absolutely. I think faking that confidence sometimes helps too. When you walk into a presentation or something where you have to put your work on display, just pretending that you're feeling comfortable is something you have to do.
Absolutely. I've been giving talks for 20 some years and I'm still nervous anytime I get up at a podium, and it makes me feel better to realize all my colleagues are as well, even those who are in their 70s. So It's a matter of mind over matter.
Yeah. So now you are a leader figure in your department. You're chair of the department, as we mentioned in the introduction. Have you found that this is a struggle that you're still having now?
Oh, absolutely. I certainly was not seeking being chair, and the people around me really encouraged me to take the position. If you were to ask me, I would have said I wasn't up to the challenge. And so far things have gone well. But I think part of it is, again, sometimes pretending or faking that you feel more confident than you really are, because others around you have confidence in you.
Absolutely. Well, it must be reassuring for all of our listeners out there who are going through this same experience themselves to know that someone in a senior position like you is still having some of these doubts and still having some of these struggles walking out on that stage for a presentation. So thanks for sharing this.
One thing I would say is one of the things I find most frustrating in this field is people who are exceedingly capable and competent who quit, who decide to not continue as scientists because they don't feel capable. And to me, that's very sad, because I've seen people who are tremendously capable not have the confidence in themselves. And what I hope all of your listeners hear is to not give up.
Well, wonderful message to give our listeners, Kristen. And I'm curious to hear about the people who might have inspired you or kept you going when you wanted to give up. So is there a particular mentor or role model you want to tell us about?
I think there are many, many role models. I'm not sure I could name one. Certainly, I've been very privileged to work with many just tremendously fantastic scientists, people who are really at the top of their field and are doing phenomenal work. That certainly has been inspiring to me, but in some ways, people who are quite a bit more advanced than I am, it's hard for those people to inspire me, because if anything, they fill me with fear. Mm-hmm. When I was a grad student, I couldn't see myself as a full professor or a chair. That was too far in the future. The people who really inspire me are more immediate colleagues, the people who are maybe a step or two ahead of me. And I'm closer to and can both provide examples for me of how it's doable to take the next step from grad school to postdoc, from postdoc to being a professor, et cetera, to write that first grant, to publish that first independent paper. And then again, people who are good enough friends to encourage me when things get tough. One of the things I really enjoy about science is it is a community and it is a family in many ways. And that sense of encouragement is, I think, extremely important for all of us to go through the ups and downs of this field.
Absolutely. And having those people going through the ups and downs with you, I think really helps along the way, so you don't feel quite so alone.
Exactly. One is up when the other is down, and you help each other.
Right. You level everything out. Yep. So now, can you tell us, where did your interest in science start, Kristen? Is there a particular memory, maybe in your early days, when you first had that interest sparked for you?
So I come from a family of scientists. My father, my uncle, both grandfathers were all research scientists of one kind or shape. So that interest in science was always there, but where it really became where I knew what I needed to study for my career was actually a class I took as a freshman or sophomore in college, which was an introductory molecular biology class. And in it, we were learning about the concept of gene regulation and particularly transcriptional regulation. And I just remember walking out of that classroom with my mind blown that cells that were smaller than you could see had machines inside of them that were smaller than you could see, that were able to know about their environment and manipulate gene expression through very intricate and logical mechanisms. to exert control over gene expression. And I just couldn't believe that such highly tuned machines were inside of us. And from then on, the concept of gene expression and how genes are controlled in different cells and different conditions has really driven all of my research.
Very cool. I think that's one of the things that initially drew me to biology, as well as this idea that all of this is going on inside me right now. It's very relatable.
Yeah.
So now, can you tell us, having this family that was engaged in science, was there a lot of pressure for you to pursue a scientific career, or was this something that you were able to decide on your own?
There was no pressure. I think I was very fortunate to come from a family where the only thing there was pressure for was to find your passion. And I was told over and over again, it didn't matter what I did as long as I loved doing it. And I saw that in the family members who were scientists, as well as other family members who were teachers or other careers. The concept that your career is not a job, is not a paycheck, but is really a part of your being, your personality, and what drives you in life.
Very cool. So is this something you then bring now to the lab to mentor your students today?
Yeah, absolutely. I think science is a field where it's very hard to be in if it isn't a passion. People who are in science, that's what we do. That's who we are. You can't really imagine not doing it.
Absolutely. So can you tell us then how you went from this curious undergraduate student then to where you are today? What are some of these key moments along the way that really got you to where you are?
The key moments have been working with people and in labs and having mentors and people support me again, as I've been saying, that as an undergrad, shortly after this class that I just mentioned, I started reading more about the field of gene expression and realized that one of the leaders at the time in the field, a professor named Mark Patashny, worked at Harvard where I was studying. And I was lucky, fortunate, stars aligned, that I got the opportunity to do undergraduate research in his lab. And that was transformative because having the community of students and postdocs there who were doing research, were learning new things every day, were excited, were passionate, were supportive, became good friends and colleagues was addicting. Again, I couldn't imagine not continuing to do it. So from there I went, and that gave me a fantastic grounding in research and launched further studies when I went on to grad school and then to postdoc.
Very cool. Now, did you do your PhD work with the same laboratory as well, or did you mix it up a little bit?
No, I didn't. I had gotten the advice that it would be good to diversify and learn something else. Mm-hmm. I had initially thought I might stay in the same lab, but the project sort of took a turn, as projects do.
Of course.
To be perhaps not as interesting as I thought the science might be going initially. When I had the opportunity entering grad school to investigate other things, I did, and that's where my research turned from this question of transcriptional regulation to the question of splicing regulation, which is really the next step in the processing of genes. And at the time that I was entering grad school in the early 90s, the question of how splicing was regulated was just beginning to be recognized as an important question. And it was a very exciting time to come into the field, and it caught my attention, and I haven't left that field since.
Absolutely. So speaking of some of these things that you're very passionate about and some of the questions that you're answering, is there a particular project, Kristen, that you have going on in the lab right now that you want to tell us more about?
The big question in the lab is how this process of splicing is regulated in the immune system. So in response to various immune challenges, whether they be pathogens, cancer, viruses, etc. And so we have a number of different projects. It's hard to pick one. In general, the questions that we're trying to understand are when you have an infection, when you have some immune response happening, how does this process of splicing change to allow your cells to respond more effectively to fight that virus, to fight that pathogen or that other immune challenge? And we're always learning something new, and that's part of the thing that is motivating in this field. We've discovered ways in which viruses, when influenza, for instance, flu, infects your cells, how the virus uses some of your cellular proteins to carry out its own splicing, which it needs to do to live. But now we're looking also at how that impacts the gene expression of the cells in your body and what that may mean for the ability of your cells to fight off that flu infection.
Very interesting. I guess are there overlapping elements of the response for a bacterial pathogen as you would see in cancer, or are they pretty distinctly different?
That's a great question and one we're trying to figure out right now. We've identified a few pathways that are involved in T cells in regulating splicing in response to, say, a bacterial pathogen. But some of my colleagues here at University of Pennsylvania are looking at those similar pathways as they relate to cancer. And so we're now collaborating with one another to ask whether the things we have learned in T cells are also relevant to regulation of splicing in cancer.
Very cool. So, Kristen, what is the next step then? Maybe once you figure this out, what will you do with that information?
There are a number of things. So as a basic scientist, my primary interest is discovery. And there's always more questions. So when we answer one, we immediately are thinking about the next set of questions. But at the same time, we're interested in hopefully using the information we learn to improve human health. And one thing that's been extremely exciting for the field that I'm in, this field of alternative splicing, is the recent approval about 6 months ago of the first FDA-approved therapy that uses modulation of splicing to fight disease. And in that particular case, it's the disease spinal muscular atrophy, or SMA. But that has been motivating for the whole field because we realize now there is precedent that as we understand more about the mechanisms by which cells carry out this regulation, that it is really possible to use that information for therapy. So whether my lab will ever actually be involved directly in that, I don't know. But certainly I am hopeful that what we learn can be used as tools to manipulate or correct splicing defects that may be contributing to human disease.
Very cool. Well, this is really exciting research, Kristen. We've talked about some of the highlights of your career path, but we haven't yet touched on a lot of the challenges and the struggles and the failures that I know are there. Many people sweep them under the rug. But I think these are really important to talk about to give our listeners the holistic picture of what life is like as a scientist. So do you have a favorite failure story you want to share with us today?
Well, I would actually say that science is basically one chronic failure after another.
I like it.
I think that's also an important concept to get across. And I actually didn't realize this until I watched a former student of mine struggle with it. That really, as scientists, we just go from one failure to another. And I think that people who really succeed in this field don't see those as failures, but see them as part of the challenge of discovery. And what is motivating is the journey and not only the result. So any given day, one experiment fails, maybe one succeeds, but then the next one fails. And you just have to keep getting up and keep going. I remember as a graduate student, I was always frustrated when I went to the professor I worked for, a wonderful scientist named Tom Maniatis, and I would come to him with a result that I was excited about. And for about 10 seconds, he would be excited. And then he would say, so what are we doing next? What's the next experiment? And I thought, just let me be excited for at least 30 seconds.
Right. Can we celebrate for like today? Yeah.
But I realize now that I've taken on that same attitude, that part of the excitement is where will that result take us? What's next? What's the broader impact? We're just wired that way to always be asking the next thing. So I think science is a bunch of many little failures with occasional temporary successes, but then you're right back into what don't I know? That being said, I will say that there are times where all scientists, myself included, have struggled with, are we really up for the challenge? And sometimes it happens at odd points. One of my biggest struggles came right after I got tenure at the University of Southwestern Medical Center, which was a wonderful institution. And tenure is an exciting thing, and it's one of those successes that occasionally happens. But it was temporary because I realized immediately that tenure didn't mean much if I didn't get a grant renewed and continue to publish, etc. And at the time, my grant was up for renewal, and I was having papers that were being rejected. And I really wondered, could I keep going? I'd been pushing for that tenure decision, but now I had continued hurdles in front of me. And again, it was really the friends around me and the colleagues around me that had been through similar things that helped encourage me to keep going. And sure enough, the grants got renewed and the papers got published, and you keep going.
Absolutely. I think that support network is key to getting through some of these tough times that we mentioned. So very cool. We mentioned some of these intermittent successes, and I think these are so important to take the time to celebrate, even if it's only 10 or 30 seconds at a time.
Exactly.
Do you have a favorite success story that you want to share with us today, Kristen?
Oh, success story.
I think these are easy to forget in the midst of all the problems that you face, but we have to celebrate them.
They are. But one thing I will say is that my lab has a tradition I really like, and that any paper that is accepted for publication or grant that is funded or student qualifying exam that they pass or thesis defense, we celebrate with champagne or beer, or if the student doesn't drink alcohol, soda or whatever. And we keep those bottles and we label them. So above my desk in my office, as well as in the student office outside, we have a shelf of many, many different bottles, all of which have a piece of tape on it noting what thing was being celebrated. And when I moved from UT Southwestern to University of Pennsylvania, the movers actually asked, they thought jokingly, whether they needed to pack all these empty bottles. And I said, absolutely.
Those are my trophies, yeah.
It is true in science that we have lots of failures. So when we have a success, we need to really celebrate it and remember that. There are times now when I get a paper rejected and I think, will I ever get another paper accepted? And then I look at the row of bottles and I realize, okay, I've been through this before. I've made it through the hurdle. We'll do it again.
I love that. So do you remember what bottle it was that you celebrated your tenure with?
Yes. In fact, it's sitting right behind me.
It's right here.
And I think there are 2 bottles of champagne for that one.
Well, you earned it. You earned the 2 bottles.
Yes.
Well, congratulations on the 10-year success, and also these little successes along the way that you mentioned, like qualifying exams, thesis defense passes. And I think all these things are really important. And I love that idea of having the physical reminder that stays in the lab and stays in your office. So now we talked about some different aspects of your life, both inside and outside of science. And one thing that I do love to chat with all of our guests about is books. So Kristen, do you have a favorite book that you want to recommend for myself and our listeners today?
I have to admit, I don't read very much. I have never been a big reader, and my work schedule, the way it is now, I really have very little time to read. So what I do read and what I have enjoyed is I have 2 teenage children, and I, throughout their childhood, read books with them, whatever they're reading. They're both pretty avid readers. So when my son was young, we went through the Harry Potter series together. More recently, we went through the Hunger Games series together. This summer, I read the Divergent series with my daughter. And that's been fun because all of those books are fun escapes from my work life and also, I think, have some good lessons in them. But it's also fun to be reading a book that one of my teenagers has just read, so we can talk about it as they're thinking through what they've learned from the book.
Yeah, it's like a family book club. That's awesome.
Exactly. And then I also get their inside jokes when they try to relate something.
Right. The cultural references are not lost, yeah.
I'm not quite as clueless as they think I am.
Gotcha. Well, I feel, I know with the schedule, it's tough to squeeze reading time in. So that's something we're trying to build a list of things for our listeners to find out there that they can read. So we'll add these different series to our list, of course. And Kristen, we've talked about some different aspects of your career. And one of the things that I love about the careers in science are some of the opportunities that you get to travel. This can be to go to conferences, to see colleagues. So is there a favorite place that you've been for science that you want to tell us about today?
There are actually 2 places that are really memorable. The first is one I went to 10 years ago, which was a town called Bariloche in Argentina. It's the northern part of Patagonia in the Andes. And I went there for a fairly small conference around the splicing field. And we all first flew to Buenos Aires, and then the conference organizers, who were some faculty who live and work in Argentina, arranged a charter flight for all of us to go to this place in the mountains. And it was just stunningly beautiful, these snow-capped mountains and beautiful crystal blue lake. And It was a fantastic meeting for two reasons. The science was just incredible. So you wanted to be inside hearing these great scientific progress and stories about research that others were doing. But at the same time, you really wanted to be outside because it was just one of the most beautiful places I've ever been to. And thankfully, we had some free time to do both. But that was a place I never would have gone to on my own and really was just strikingly beautiful.
I've heard wonderful things about that area. It's quite gorgeous.
It's just stunningly beautiful. I've been to the Swiss Alps, and those are also gorgeous, but this was maybe even better.
Did you get to go hiking out there?
We did a bit of hiking. We also took a boat trip across quite a large lake and did some just small hiking around as well. We didn't go very high.
Wonderful. And now in the second place you were going to mention, go ahead.
The second was a trip more recently, last year, to Japan and Singapore. And that, again, was for places I might not have gone to otherwise, but I had back-to-back conferences there. And my son had also just graduated from high school. And so what made that trip very special was that I brought him with me as part of his graduation present. So the chance to share some of my work life with him, as well as we took several days to just be tourists and see Tokyo and see Kyoto and see Singapore, was really fantastic. And actually it made a big impression because my son is now trying to get back to Singapore next summer for a work internship in college.
Very cool. So are your kids interested in science as well?
No, absolutely not. No. Neither of them like science at all.
That's fair.
They both are fascinated by history and politics and other wonderful things, but they're definitely making their own path.
Very cool. So what were some of your favorite sites in Japan or Singapore?
Everything was interesting and exciting. I think seeing a lot of the temples in Japan was really striking and something you would not see, at least in the places I live in the US. And in Singapore, what I was most struck by was how green many areas of the city are. I mean, it's a small place, but they've managed to work in some beautiful nature, even within or on top of the buildings. I was really struck by that.
Yeah, that's really cool. Well, thanks so much for sharing some of your travel memories with us today, Kristen. And we talked about one of the traditions you have in your own lab, and I love the idea of keeping the bottles from all these celebrations. And I think it's fun to talk about some of these traditions or funny memories or fond memories that people have in science, because I think not everyone realizes that science is a social endeavor and that scientists maybe don't meet all of the stereotypes that are portrayed about us in the media. So do you have another maybe tradition you want to tell us about that you experienced in a previous lab, or just a funny memory that you can share that really shows this human side of science, that we're not little robots working in labs?
Oh, we're definitely not robots.
Mm-hmm.
I think that's one thing that maybe doesn't get enough credit is that scientists are people and we have families and friends and lives. So working with one another, you also become close and become friends. And the biggest lab tradition is the one that I already shared. We also try to every summer do a lab outing. The tradition for many years has been to go to a baseball game. That's something that labs that I was in as a postdoc and grad student also did. And I think it's important to spend time socializing together as a lab as well as working, because it is a very collaborative field and we do better science if we get along with the people we're working with and can share ideas and share reagents and share help through the experiments. So building a community, I think, is really critical to doing good science.
Definitely. I know every lab I've worked in, you sort of get that family vibe when you go in.
Yeah.
Everyone works together and everyone's a little bit different, but everyone's got their own preferences, but you all work together towards the same goal, which is really cool. So now you mentioned baseball games. Are you a baseball fan yourself?
I'm a sports fan. Baseball is a convenient excuse to go have a beer and hot dogs and sit outside. But football is a sport I most like to watch, and soccer is the sport I most like to play.
Gotcha. So do you still get to play soccer these days?
Only when my kids' soccer teams have parent-kid scrimmages.
Oh, that's awesome.
Which is always a unfortunate thing because I hurt afterwards.
Yeah.
So mostly I'm a soccer spectator now.
Well, great to hear a little bit more about some of these traditions that you have in the lab. And I love the idea of getting out and socializing and spending time with your group outside of the lab. I think that is really important because you are working together to answer some awesome questions that we talked about earlier in our interview. But there's always something standing in the way, Kristen, I've found, whether it's funding or staff or technology or feasibility, something's just keeping you from answering that question that you are dying to know the answer to. So if you could have anything, if you could have all the resources you needed, what is the question that you would want to tackle?
The question that I'm most fascinated by is actually one that currently we're not focused on much at all, largely due to technology issues, but which is mechanistically how this process of splicing occurs. So fundamentally, I'm a biochemist. I am turned on by how molecules get in with each other and influence each other's behavior. And we know an increasing amount of how splicing, how this process of cutting out parts of the instruction manual occurs chemically. And we know the basic enzymatic complex that carries that out. And that's a complex called the spliceosome. And in the past 2 years, there have been phenomenal breakthroughs in high resolution structures of the spliceosome using cryo-EM technology. So we now know a huge amount about what that machine looks like that cuts and pastes the RNA together. What we still don't have a great understanding of is how that process is regulated. We know the players, we know the proteins that can influence where the spliceosome carries out its activity. But to a large extent, we don't know how those players, how those proteins work. My slides, when I give a talk, show one small circle next to a big circle being the spliceosome. And sort of the implication is the protein binds and then magically it promotes or inhibits the use of splicing. And I would love to really know how that happens. We know some big picture answers to that question, but we're really still missing a lot of the details.
Gotcha. Do you think it's a technology limitation that you're not able to see what's happening at that small scale? Or
So the real issue is that most examples are probably different from one another. That is, there isn't just one way this can happen, but many different ways. So capturing enough of a single complex, a single event to be able to do high resolution structural studies or biochemical studies is really the limiting step. So if we can get a lot of any size complex, we can get a high resolution image of it now. But this process is very diverse and very transient. And so figuring out how to capture it and build up enough of that material that we can really see what's going on is the hard part.
Gotcha. Sounds like we need to build up your lab army, so you need more hands to do all the work, right?
Well, I think it's something that's really going to come from the community working together, and that is each of us.
Sure.
One person may have a technology, another person might have the appropriate system, and then putting that together.
Very cool. Well, Kristen, thank you so much for thinking big with us today about these questions that remain unanswered in your own field. And we chatted about some different aspects of your career throughout our conversation today. And one thing I love to leave our listeners with is some advice that they can use on their own career journeys. So, Kristen, is there one thing that someone told you along your path that really helped you that you can pass on to our listeners today?
I'm not sure there was one piece of advice. I think the encouragement that people gave me made a tremendous difference. The advice that I can make it, I can continue in this field. The specific advice that I most remember are what various mentors taught me about writing. Don't ever use the word can in a grant because it means that you're not going to actually do it. Don't use the word this without describing what it means because nobody else is going to understand what seems obvious in your head. Those are the advice that I most go back to. Communication is critical in science. Being able to communicate clearly and succinctly is the key to getting grants, to getting funding, to communicating your work in a talk. So those are the pieces of advice that I think have had the biggest impact on my ability to be successful.
Certainly. And I think science communication is big on a lot of people's minds right now. So in your experience, is there something that really helped you develop those communication skills other than these little tidbits of advice that you received from people?
Again, I was fortunate to work. Both my undergrad and graduate mentors, who I've already mentioned, are excellent writers and communicators. And I had the privilege of learning from them even to the point of the first things I wrote as an undergrad and as a graduate student. They would go through very meticulously with me, sentence by sentence, and really not just took my work and edited it, but taught me why one way was better than another. And that was tremendously time consuming for them. And I'm not sure I would have that same patience if I had been them. Mm-hmm. But it really made a big impact on me.
Very cool. Well, Kristen, thanks so much for sharing this advice with our listeners. Is there any other last piece of advice or a last message you'd like to leave them with today?
I think the thing I hope people hear in everything that I said is that science is a fantastic career. It's a flexible career. I think often people, especially women, get worried that you can't have a family, have kids, if you really want to make a career as a successful scientist. I think that's really incorrect. I'd love the ability to work my experiments around when my kids might have a soccer game or a school play or something. And it's not as scary as it seems. And those of us who have done well are not any more capable than most other people. We just sort of refuse to give up. That's what I would hope to leave with the listeners, that if this is something they love and are good at, that they shouldn't give up. And it won't always be easy, but it's worth it.
Well, wonderful words of encouragement. Listeners, you can do it. Don't give up. That's awesome. Now, Kristen, can you tell our listeners if they want to learn more about your research or more about you, where should they go?
Probably the best is to visit my lab's website, which is www.kwlynchlab.org. And that has contact information for me and pictures of lab outings at the ball game and lab graduations and a description of our research and recent publications.
Fantastic. Well, listeners, check out the website, learn more about Kristen and her research. And Kristen, thank you so much for joining us on the program today and sharing a piece of your story.
My pleasure. Thank you very much for the invitation. It's been a lot of fun.
Absolutely. It's been great to chat with you. And listeners, wonderful to have you here as well. We'll see you next time on another episode of People Behind the Science. Your voyage to explore the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to chatting with you next time on People Behind the Science.